In this video, I want to do the test review for Chapter 17 on nucleic acids. First question dealing with that is the second one here. Now it's asking for the steps and names and different enzymes for the synthesis of proteins starting with DNA. So this question here is for me to be able to cover all the different steps in the process, the different terms and different enzymes, etc. So let's start out with DNA, right? That's where all the genetic information is stored. And proteins are not, formed directly from DNA. First, something called messenger RNA needs to be synthesized. And that process is known as transcription. And the enzyme responsible for that process is called RNA polymerase. The messenger RNA is carrying the genetic code, basically, right, for the synthesis of the protein. And then the protein is finally synthesized in a process called translation. And the enzyme that's responsible for that is called the ribosome. And just to be clear here, the ribosome is different than just a traditional enzyme. A ribosome is composed of RNA pieces as well as protein subunits. The technical name, which you don't need to worry about for this class, is a ribozyme. Now, these are the basic steps for, from the genetic information to the protein. There's one other thing that I want to just talk about briefly here, and that's the issue about the synthesis of DNA, right? That's another important process that we covered in Chapter 17. And so when DNA is synthesized, when it's replicated, that's replication. And there's two enzymes to be familiar with here. One is the DNA polymerase, which is carrying out the bulk of the work. And then a second one is called helicase. Now that's just a general term for this particular, let's say, class of enzymes. The helicase is what's going to unwind the DNA. Remember, the DNA is double-stranded. It needs to be opened up before the DNA polymerase can get to that information to then replicate. Now we've seen one type of RNA here, messenger RNA. There's also a type of RNA called ribosomal RNA that's in the ribosome. And then the last type to be familiar with is this type, where it brings amino acid to the ribosome. This is called the transfer RNA. In the next question, right, there's two different types of nucleic acids that we see in nature. And we want to know sort of the basic structural features that distinguishes one from the other. Now, a couple of the obvious ones. One, ribonucleic acid. Well, this has ribose as a sugar. And then deoxyribonucleic acid. Well, this is deoxyribose. So these are two very similar sugars. They're both aldopentose sugars. Okay, and the deoxyribose sugar has the hydroxy group is missing on the number 2 position. And I'll talk about that a little bit more in a minute. Now, another structural feature that's really commonly referenced here is the difference in the nucleic acid or the nitrogenous base sequence here. So in messenger RNA or in RNA, you have A, U, C, and G. So uracil replaces thymine. And then the last sort of commonly referenced structural feature is that your RNA structure is going to be a single-stranded piece, and then the DNA is a double-stranded polymer. Now, the single-stranded piece does fold up on itself, but if you grab a 3' end and a 5' end and you pulled it apart, it would be a single strand. There's a decent picture of a transfer RNA in the textbook that can illustrate that single-strandedness and sort of how it's folding up. Whereas the DNA has two separate strands, and then they come together right in the anti-parallel fashion, and they form the double helix. Now, if we look for the other questions, the next one dealing with Chapter 17 is this one here, number 9, for drawing the messenger RNA structure. Now, in this particular problem, I'm saying that this is an RNA structure, so then, we see uracil, right? And then we would also be using the ribose sugar, and we'll see how all those pieces get put together in just a minute. The other notation that we often use here is that, if it was a DNA structure, what you see is a lot of times is a lowercase d in front of the sequence. Right? Now, given the sequence, you would also know it's DNA because you see thymine instead of uracil. And so that's one of the giveaways with respect to whether you're looking at RNA or DNA. Now, there's another piece of information that you should be familiar with, and that's the orientation of the sequence here. So whenever you see a nucleic acid sequence, whether it's RNA or DNA, we're always referencing the 5' end first and then the 3' end. And remember, those are referencing the carbons on the ribose sugars. And we'll revisit that once we start drawing the structure here. So whenever I'm drawing these structures, what I usually get out of the way first is my phosphate sugar backbone, okay? And usually I draw this as a little angle. Now, if I was drawing the normal ribose sugar, there's a particular pattern for the OH groups, fine. But in the context of a nucleic acid structure, okay, one thing to know is that on this top, and by the way, this is going to be the 5' end here, and this is where the uracil is going to be bonded. And I'll draw that uracil at the end. So let me go ahead and get the numbering out of the way. So this is going to be the 1' carbon, 2', 3', 4', and the 5'. The reason why we use a little prime on the sugars is because the nitrogenous bases have to be numbered as well. And for the 1, 2, 3, etc., for those bases, there's no prime associated with it. So it's my understanding that they chose to put the prime on the sugars, and that's what we use. Now, we only need to know this numbering. We don't need to know the numbering for the nitrogenous bases. Okay, 5' end. One thing to know about is that there is a phosphate group that's going to be sitting up here. I'll talk about charges in more detail in just a second, but I'm going to go ahead and add them here. Put circles around it just to emphasize it's a charge. And one thing we learned is that the connection of the phosphate sugar backbone is through the 5' and the 3' position. So what happens is that when you're drawing out the backbone here, it alternates a ribose, a phosphate, a ribose, a phosphate, a ribose. Okay, so let's do that. So it's not an OH group sitting here, right? These OH groups are being used to bond with the phosphate group, and the phosphate group act as a linkage between the individual sugars here. So in my label here, I have 3 bases. So I need 3 sugars, 1, 2, I need 1 more sugar. And again, that pattern, right, the connectivity is going to run through the 5' and the 3' positions. It's not a hydroxy group, it's another phosphate. And just to switch it up here, just to kind of emphasize something, it really doesn't matter, which position you put the double bond, as long as you don't place it, where the connectivity is. Right? Whichever oxygen is freed, you can put the double bond. And now that I've had my 3 sugars, now the 3' end will be a hydroxy group. Now, since I'm dealing with RNA, ribose, then at the 2' position, there's also a hydroxy group. Let me add those. The charges over here. Okay, so, your typical phosphate looks like this. What we learned about in 151. So basically it has 3 positions that could be used to bond with something else. And every time that oxygen is bonded to something else, it loses the negative charge. So like here, since I have, 1 bond here, 1 bond there, there's room for the double bond, and then you have a single negative charge. And the same thing down here. And again, it does not matter where the double bond or the single bond go. You can alternate the two. It really doesn't matter. We don't talk about this topic in this class, but the pi bond and the negative charge, they're delocalized through resonance between these 2 positions. So that's why it really doesn't matter where the negative bond or the negative charge shows up. And really, the last thing to do is then to go in and add our bases. So they're going to be connected at the 1' position for each of the sugars. And again, uracil is going to be here, cytosine, and then adenine down here. Now, for this test, for this class, you'll be given the structure. So let's take a look at those. There they are. And it's basically this handout that you'll be given. And what you have to realize is, the way that these structures are presented, it'll be the nitrogen at the bottom here that's going to be connected to the sugar. And then on the purines here, it's the nitrogen that is on the small chain or the small ring structure that's going to be connected to the sugar. And that goes for whether it's RNA or DNA. And so now I'm going to go ahead and redraw this, and I'll skip forward on the video. Okay, so there's the completed structure. And again, you can check my work if you want. That's the uracil, that's the cytosine, and then that is the adenine. The other thing that I would like for you to know is that the bases with the single ring, these are referred to as pyrimidines, and then the doubled ring bases are called purines. And I think I'll write that on the handout here. So again, these are pyrimidines, and then these are the purines. For what it's worth, the way I remember this is that the larger structure gets the smaller name. By the way, if I was going to compare the messenger RNA structure versus DNA structure, so the one big difference would be that you wouldn't have uracil here, you would have thymine. And then the other thing with the deoxyribose, there would be no hydroxy group at the number 2 position. So structurally, those are the two things that you would see that's different if you had to draw an RNA structure versus a DNA structure. And the last couple questions for chapter 17 is looking at some a DNA structure and asking you to sort of walk through the process of transcription and translation. So one thing that you'll want to pay close attention to is with respect to which side of the DNA is being presented to you. Okay, so in this one, this is the template strand. This is the portion that the RNA polymerase would actually read and then convert to the messenger RNA sequence. And when you do that, basically you're playing the role of RNA polymerase here when you're given the template strand. Let's go ahead and do that, and then I'll address the other half of the DNA and what you would do if that was given to you. Okay, so this is a template strand. I'm playing the role of RNA polymerase. And what I'm going to do here is I'm going to use complementary base pairing between the DNA strand, okay, and then the new messenger RNA strand. And then what you have to realize here, and let's put that over here. So in the DNA, what the possibilities are T, A, G, and C, going from DNA to messenger RNA is complementary base pairing. So the complement to T, if you will, is A. The complement for A, and this is the one you got to watch out for. In DNA, if we were dealing with replication, we'd say A to T. Okay, but there's no thymine in RNA. Instead, uracil replaces thymine. So if you see, right here, if you see an RNA in the DNA template, then you're going to have a uracil in the messenger RNA sequence. And then down here, you have the typical base pairing. G to C, C to G, that's always the case, no matter which pattern you're going through. Okay, so let's go ahead and do this. So again, and it's not a bad thing to put this little, it's like a little cheat sheet here for you. So I got 3 T's showing up. That means in the messenger RNA I have 3 A's. A in the DNA, that's going to give me a U, and so on and so on. So there's my new messenger RNA sequence. And again, this would be something called transcription. Now for this problem, I don't want to stop here. I want to go to the next step. Now for the next step, the translation part of things, well, now you're playing the role of the ribosome, okay? And when you go from messenger RNA to then using genetic code to figure out what the protein sequence is, you don't just start at the beginning of the messenger RNA sequence. The place where you start is the start codon here, AUG. Let's look at the genetic code just for a second. Okay, so now we're going to be using the genetic code, and this will be given to you on the test, okay, to go from the messenger RNA to the protein sequence. Most of the codons code for some amino acid. But there are a few here that code for start, and well, 1 codes for start and then 3 code for stop. And there's our AUG. That's what you always look for when you first start figuring this out. And this gives you the reading frame. Right? So if the ribosome is going to start here and it's going to read 3 at a time, then those are your codons. These are the ones you're going to use in the genetic code to then figure out what protein sequence you're dealing with. Let's do that. Now for AUG, you can put methionine or start, doesn't matter to me. Next one is AUU. Let's just practice a little bit here. So there's A, second letter U, there it is, AUU. So isoleucine is the first protein or first amino acid in our protein. And then I'll let you check the rest. AGU is serine, then it's followed by tryptophan, arginine, and threonine. And that's it for this sequence. Okay. Now there's no stop sequence in the little chunk of messenger RNA that we're using here. That's fine. You know, we're just practicing going through our complementary base pairing based on whether you have the template strand or whatever here. Right. In other cases, if I hit a stop codon early on, then I stop and I don't translate anymore. But for this particular example, here is my fully translated messenger RNA, again, from my original template strand. Let's talk about the template strand just a little bit. Okay, so what you see in purple here, that's if it's indicated that you're dealing with a template strand, you use a complementary base pairing to figure out what the messenger RNA is, etc. The other label that's used here is the coding strand. Now, the newer edition of Timberlake is using the informational strand, I think, or informational sequence. Now, I'm not going to do this full thing here because it'll just be confusing, okay? I mean, this stuff, when you first get a hold of it, can be confusing enough. If you're given the coding strand here, you don't go through this stuff. Okay, really, all you're doing, if you're given the coding strand to figure out what the messenger RNA is associated with the coding strand, is you just change, you don't even do complementary base pairing. All you do is you change the A's in the sequence to U's. So if that was the case, right, there's only 4 A's in my sequence. Again, okay, if I'm pretending like this is the coding strand, and all I would do is literally change these 4 A's to U's and keep everything else as is. Keep the T's, the C's, the G's where they are. And then coding strand, then for doing that on the coding strand would then give you that messenger RNA, which would be a separate messenger RNA than what you're seeing here. Just keep that in mind. Again, the full description here, see the textbook, see the videos. I'm not going to do it here because it'll just be messy. And, you know, not to muddy the water unintentionally, okay? There is another type of complementary base pairing that we discussed, and that is between the messenger RNA codon and then the anticodon that's on the transfer RNA. And let's just take one of these as an example, just this tail end here. So again, this would be the messenger RNA, and you kind of have to imagine the scenario here. You have the ribosome holding the messenger RNA, if you will, and there's a little slot where the transfer RNA is going to come in and bring the appropriate amino acid. Okay, so there's a transfer RNA that's going to bring in the threonine here. But the threonine does not interact directly with this codon. Instead, it's this large transfer RNA molecule here with threonine poking out. Let me try to draw that. It'll be messy, but that's fine. So imagine in blue here is my transfer RNA, and just forgive me. Just let's make it a little squiggly. Let's try that. Okay, so again, imagine that this little blue squiggly thing that I'm drawing is the actual transfer RNA. Up top here is something we call the acceptor stem. This is where the threonine is going to be positioned. Okay, and when the amino acid is attached to the transfer RNA, we call that a charged transfer RNA. And then down here on the bottom of the structure of the transfer RNA, you have the anticodon. And there's complementary base pairing here between the transfer RNA and then the messenger RNA. And actually, let's just run through that like we did up here. And actually, I think I'll squeeze it in right here. Right, if I'm going from messenger RNA to transfer RNA, well, what are my bases? I have these bases. And now the complementary base pairing is A to U, U to A, C to G, G to C. Like I said earlier, C's and G's always go together. A's, T's, and U's are the ones you got to worry about. If it's RNA to RNA, then it's the AU-UA complementary base pairing. It's only going from DNA to messenger RNA where, since we don't have thymine U is inserted in place of thymine. How does this complementary base pairing work here? Well, if I wanted to know what the anticodon for this codon is, well, again, complementary base pairing based on this information. If my messenger RNA is A, then my transfer RNA would have a U up here. C and G is always complementary to each other, and then A and U. So this sequence here, UGA, is referred to as the anticodon. This is how the ribosome builds the proper amino acid sequence up. It's using this, triplet codon language and the genetic code to then bring in the proper amino acid one at a time, based on ultimately the complementary base pairing here. Last chapter 17 topic, at least on the test review. So I'm laying out a mutation scenario here. So I just copy and pasted this sequence and made one little change here. And that change is right here. Right, that's the one difference, okay? You could call it a point mutation, that'd be fine. This is more of a general term. The more specific label for this mutation is a substitution mutation. And this is more specific. Point mutation can be frame shift or substitution. We dealt with the frame shift mutation in the lectures. I'll leave it there. Here, all I'm doing is that instead of a C, I've made an error, or DNA polymerase has made an error, and the error wasn't caught, wasn't corrected. And now you have one nucleic acid that's wrong, and it's something different than what should be there. T has been substituted in place of a C. Now, since that's the only thing that changed here, so my reading frame on the original one began starting with the third base up here. Okay, so the first two T's were not read, so let's kind of imagine we mark those out. This is going to be the beginning of our reading frame here, right? Because the mutation occurs downstream, so we don't have to worry about. Right? This is going to be read exactly the same as before. Okay, so there's our reading frame. If we go through this sequence again, it's the same thing until we get to that substitution. So let me just quickly fill all this in. Now, in terms of the transcription process, the messenger RNA is just going to synthesize the RNA polymerase is just going to synthesize a new messenger RNA as is. And then it's the ribosome that's going to realize that something, well, I won't realize it, but it'll just, instead of inserting, right? So if it's, there's our mutation right there. So instead of inserting tryptophan here, if I look at UGA, this is actually a stop code right there. Okay, so now look what's happened with that. A single point mutation caused a very dramatic change in the protein sequence. In the original structure, it had and again, this is just the front end of a particular gene, let's say, and it would keep going on and on. So it'd be a very large protein, for example. If I had this one point mutation at the very beginning, and again, it's only taking one position to do this, and it can completely shut off the translation, the synthesis of the protein. And sometimes point mutations cause that kind of dramatic change. Okay, I think that'll do it for chapter 17. Thanks.